Now we know about the R groups in the amino acids. Let's look at the overall structure of proteins and how they're built. So we know that proteins are long chains of amino acids that are linked together. The things that hold those monomers, called amino acids, together is called a peptide bond. This is a specialized type of amide bond. An amide, if you recall, is anything that has a C double bond O and a nitrogen. But a peptide bond is specifically an amide bond that is between two amino acids. So not just any amide, but only if there are two amino acids joined together by something called a peptide bond. Amide bonds, and therefore peptide bonds, are known as some of the strongest types of covalent bonds and are very, very difficult to break. If we need to break them, we call it hydrolysis, and when we try to hydrolyze a peptide, we usually need a special catalyst to help us hydrolyze it. So we use something called like peptidase or something like that, and it will break it, but it is very difficult to do. And after a series of steps, you'll go back to having your amino acids. Again, the process of hydrolysis does not happen readily under normal conditions, but you will have to have a catalyst to really help digest these proteins. There are two general types of proteins that are found in the body. The first type are the fibrous proteins. These are long, linear, side-by-side polypeptide chains. These are insoluble in water, and they are used for structural purposes. The second type is called globular proteins. The globular proteins are polypeptide chains that are rolled up, and they look like a glob, not linear chains. They are soluble in water, and generally most of the things that they do require that they are able to move from one position to another. So they might be attacking an intruder, they might be delivering something. Those types of things require mobility. When we look at protein structure, we see that there are 4 levels of structure. We start with primary, then there's secondary, tertiary, and quaternary. There's more information on this page. This is for you to refer back to. But we can see that those 4 levels all are related to each other. They all have different characteristics, and they are held together by different types of bonds. And that's what we will be focusing on next. The first level of order that we're going to talk about is called primary structure. Primary structure is the sequence of the amino acids in the polypeptide chain. The primary structure is maintained by peptide bonds. Those are the very strong amide linkages that we talked about. Very, very difficult to break these peptide bonds. The primary structure determines how the rest of the structure of the protein will come out. They dictate the structure of secondary, tertiary, and quaternary structures. This is similar to the way that when we put letters in a certain order, we get words out of them. It determines the next levels, and it'll tell us, well, the sentence will end up. If I don't have the letters right, the sentence won't end up right, the paragraph won't end up right, and the story will be all mixed up. The next level of structure is called secondary structure. Secondary structure is when we have a regular repeating arrangement of some kind of polypeptide chain. Within the polypeptide chain, a series of amino acids will be repeating, and when they repeat, it tends to make these special structures. These 2 special structures that we're going to focus on are the alpha helix and the beta pleated sheet. I know that the notes say 3 and the book says 3, but we only really look at these 2 special groupings, the alpha helix and the beta pleated sheet. The third one that the book mentions is the random coil, and by definition, if it's a random coil, it cannot be a regular repeating structure, so it doesn't fit in with this pattern. It ends up that secondary structures are all held together by hydrogen bonds, and what they're doing is they're linking carbons and oxygens and hydrogens that are very far apart and holding them together so that they make this very nice repeating pattern, and you can actually see a coil or a pleated sheet. The alpha helix is shaped like a spring, and it gives that property to those types of proteins. Those proteins tend to be very stretchy and snap back to their original shape. A pleated sheet is sort of like origami, where you have a series of papers stacked on top of each other and you fold them, and then they make a pattern from that. The beta pleated sheet is much firmer and does not stretch well. It tends to be more of a structural component. As mentioned, we have both globular and fibrous proteins. It ends up that the fibrous proteins contain only one single type of secondary structure. Some of these fibrous proteins are all alpha helix, and others are all beta sheet. Wool is an example of a fibrous protein that has only alpha helix. One of the things that we know about wool, it is very stretchy. And the reason it is so stretchy is because it has that alpha helix, which looks like a spring. So it's kind of like a lot of springs going and stretching and pulling. Now, wool happens to be made up of a lot of short fibers that are able to be spun together. These fibers tend to be between 2 and 10 centimeters long, and we're able to spin them into yarn, as you may have seen, which can be made into fabrics. Other proteins that have only beta sheets are something like silk. Silks are known to be very, very strong fibrous proteins. These proteins are made only of beta sheet, and because those beta sheets are stacked upon each other, they become very, very strong. This strength and stability is one of the reasons that silk was used for parachute material and is now used for silks where people hang from them. Because the structure is made out of all of these beta sheets, it is not very stretchable, which is really good if you're hanging from it or trying to parachute from a plane. Another former use of silk was to use it as a bulletproof vest. Because of its strength and its stability, again, a bullet cannot shoot through thick layers of silk. The design of silk has been incorporated into the design of Kevlar. In fact, for its weight, silk has stronger tensile strength than steel or Kevlar. And again, that structure of Kevlar is based on the structure of silk. Interestingly, while wool is made out of very short fibers that are spun together to make string and yarn, silk is very long. In fact, the silk from a silkworm is usually between 300 meters and 2,000 meters long, and that fiber is about 10 to 20 micrometers in thickness. To get an idea of how fine that is, we know that the human hair is around 80 micrometers in diameter, and that is one of the reasons that silk is so flexible and seems to flow. But silk has low elasticity. It's not very springy, and when you stretch it, it tends to hold that if you stretch it too far. While fibrous proteins have only primary and secondary structure, globular proteins have a combination of primary, secondary, tertiary, and oftentimes quaternary structure. Primary structure was the order of the amino acids. Secondary structure had some very specific patterns like the alpha helix and the beta sheet. Tertiary structure, our third level of structure, is the unique and specific pattern of folding that is found in a single polypeptide chain. So the overall shape of that polypeptide chain will be called its tertiary structure. The tertiary structure is determined by the order of the amino acids. So primary structure rules all of this, but the overall shape will be considered its tertiary structure. It does include secondary structure within it because those coils and beta sheets are part of the overall shape. And how is this shape held? Well, it is maintained by hydrophobic interactions. That means the things that hate water are found in one area and things that love water are found in another area. They clump together and we find patterns that will match that. Tertiary structure is also held together by hydrogen bonds. Again, because secondary structure is held by hydrogen bonds, it is definitely also going to be part of the tertiary structure. But the hydrogen bonds can be found in other areas, not just where there's secondary structure. An even stronger force that holds together the secondary structure is the electrostatic interactions. This means that there's a positive and a negative charge, and they're being held together by those ionic bonds. These ionic bonds are much stronger than the hydrogen bonds or the hydrophobic interactions, and they're very important to the overall tertiary structure. And finally, we have disulfide bonds. These are a type of covalent bond that is found between two sulfurs, coming from two different amino acids that have sulfur in them, We're talking about cysteine, and those two bond together, and they're held together by that covalent bond, which is a very, very strong bond. So this list is written in the order of strength. The weakest interaction that holds the tertiary structure is the hydrophobic interactions. The next strongest is the hydrogen bonds. Stronger than that is the electrostatic interactions, and the strongest of these is the disulfide bond. Those disulfide bonds are the hardest ones to break. Yet all of these types of bonds are much weaker than the amide bond, the peptide bond that we see in the primary structure. These ones can all be destroyed easier than the ones in the primary structure. So far we defined primary structure as the order of the amino acids. Secondary structure was the special patterns within, and we said that tertiary structure was the overall shape of a single strand of a polypeptide. So if tertiary structure is the overall shape and folding of that polypeptide strand, what in the world would quaternary structure be? Well, it ends up that quaternary structure is the structure that is found when there are two polypeptide strands joined together. Each of those strands are a single polypeptide strand, but they're joined together. We call them subunits of an overall protein. These proteins are not active unless all of the subunits are present. A protein that has more than one polypeptide chain is called an oligomeric protein. Therefore, quaternary structure is found only in oligomeric proteins, and it designates the characteristic manner in which the individual folded polypeptide chains fit together. So it talks about how they join together, and it gives the overall structure of the whole protein. If it's an oligomeric protein, we will find it has quaternary structure. Like tertiary structure, quaternary structure is maintained by the four types of interactions. We have the hydrophobic interactions, the loving and hating of water, the hydrogen bonds that are found in secondary structure but throughout the whole protein, electrostatic interactions, those are the ionic interactions we talked about, and of course by the covalent bonds known as disulfide bonds. Since my drawing skills leave a little something to be desired, I thought I would show you what it looks like with some computer graphics. So this is our primary structure, and primary structure is the order of amino acids, and it is held together by very, very strong peptide bonds, which is a type of amide linkage. Next, we have secondary structure. Secondary structure is regular repeating units of some sort of pattern. The two patterns that we see are the alpha helix and the beta sheet. Secondary structure is held together by hydrogen bonds. These are fairly weak inter-particle forces. Fibrous proteins are expected to have only primary and secondary structure. And here we see the tertiary structure, which we start to see in globular proteins. Remember, tertiary structure is the overall structure of a single polypeptide strand. It includes the alpha helix and the beta sheet, but then it has other random coil that is being held together. This is the overall structure. There are four forces that hold together this tertiary structure, listed from the weakest force to the strongest force. The weakest one is going to be the hydrophobic interactions, that's water-loving and water-fearing. The next strongest one is the hydrogen bond, which we see primarily in the secondary structure, but we do see throughout the whole protein. Then we have the ionic interactions or electrostatic interactions, where we have an ion, a positive and a negative, that come together. And finally, we have the disulfide bond. This is a specialized structure where two sulfurs bond together in a covalent bond. Again, that one is one of the harder ones to break. And finally, oligomeric proteins have quaternary structure. Not all globular proteins do this, but in some cases, like this example, which would be something like heme, which is found in hemoglobin, has 4 subunits. So oligomeric proteins have 2 or more polypeptide chains that are bound together. Therefore, the quaternary structure is defined as the overall structure of a protein that has subunits in it. The way that these are bound together are the same 4 forces that hold tertiary structure, and again, those are hydrophobic interactions, hydrogen bonds, electrostatic interactions, and the strongest one, the covalent bond called the disulfide bond. When we put those four levels of protein structure together, we get something called the protein conformation. The protein conformation is defined as the three-dimensional arrangement or shape of a protein. It is described by the secondary, tertiary, and quaternary structure of that protein. So the conformation describes the overall shape. But the problem is that when we purify proteins, we often have to take them out of their normal environment, and so we end up changing the secondary, tertiary, and quaternary structure. When we change the structure, it still has a protein conformation, it's just not the natural conformation. So it still has conformation, it's completely different. When we remove its natural conformation and we make it into something else, we call that denaturing. Because man-made conditions are not the same as natural conditions, it is very difficult for any proteins to be formed by man. And they do have a protein conformation, but they don't have the biological activity that they should have. The natural protein conformation that is found in nature is known as the native conformation. The native conformation is found under the biological conditions that protein is found in. When the protein is in that conformation, it can do its job. That means it has its biological activity when it has the right shape. If it doesn't have the right shape, it can't do its job. Denatured proteins are no longer active and do not work in the system. Our next video will discuss how to denature a protein and what happens when a protein is denatured.